Assalamu alaikum everyone, my name is Muhammad Dawar Zahid, and in this video we'll be talking about reproduction in bacteria. And before you watch this video, I would kindly request you to please subscribe and also to like the video. And just before I start, I want to give you guys a brief introduction, which is that so asexual reproduction, so there's two types of reproduction in bacteria. There's asexual and there's sexual. And asexual is the one that is more common. Okay, so bacteria generally go for this, like this is their normal regular mode of reproduction. Okay, so this one is more common though. And the asexual one, this is atypical. This is not something that you normally see. This is not like with meiosis, nothing like that happens. So there's a question that they love to, there's a concept that they love to ask you, that what is the source of genetic variation in bacteria? That how is there genetic variation between different, like where does the genetic variation really come from? So what I want you guys to keep in mind is that genetic variation is always going to be coming from genetic mutations. They're always going to be coming from genetic mutations. Okay, so because there's no typical sexual reproduction, there's no crossing over, there's no random assortment of chromosomes, there's nothing like that happening. So reproduction in bacteria is only the source of genetic variation is through genetic mutations. And the reason why this is also effective is that reproduction in bacteria is generally very fast, plus the genetic material is not very stable. Okay, it's not as, I wouldn't call it unstable, I'll just say not very stable. Okay, it's not as stable as your eukaryotic genetic material. So it's not that stable. You don't need to know why or how it happens, but just keep this thing in mind, because this is a concept that I've seen them testing quite regularly. Okay, let's move on and let's start with asexual reproduction. Asexual reproduction, I've already written, it's pretty common. And so there's two methods. There's going to be binary fission, and along with binary fission there's also going to be budding. Okay, it's sort of similar. Like I remember when I was giving my MDCAT and all of these different exams, it was quite difficult for me to really differentiate between these two, but I'll explain it to you guys. Okay, so obviously it's asexual, so it's going to be genetically similar. I'm not going to be writing that, that's pretty simple. What really happens is, let me just quickly make a bacterial cell. So what happens is that the DNA is going to be going here, it's going to go to the cell membrane, it's going to get attached to the cell membrane, and when it's at the cell membrane it is replicated. Okay, so it becomes, sorry, just a second, why did this thing even happen? Let's just, oh my god, okay, so let's just put it here. So it becomes replicated. Okay, we've done that. Now what happens is that this entire cell is going to be enlarged. Let's say it becomes this big. Okay, it becomes this big. Now a cross wall is going to be forming between these two pieces of genetic material, right? So this happens. Now what happens is, this entire thing grows, and then it becomes separated. So this thing will become separated, this thing will become separated, and you will have two cells. Okay, this is what happens. Please just try and understand this conceptually. You don't really need to remember this. I'll just quickly give a recap of what is happening. The genetic material, it basically sits in a corner, becomes replicated. The cell is then going to grow, and then a cross wall is going to form in the middle, and then these two cells are this big cell is going to be separated into two daughter cells. Okay, two daughter cells. That is what is happening. And one thing that I want you guys to know is that binary fission, the normal rate of binary fission is once every 20 minutes. Okay, so just keep that thing in mind. And aside from that, it's pretty simple. There's nothing to worry about. And now let's move on to budding, what budding really is. Okay, so let's do the same thing. Let's make a bacterial cell over here. Okay, so in this bacterial cell, what is happening is that at one end of the cell we have this thing, this sort of like this protuberance, sort of like this protrusion. And in this protrusion, what we have is that, let's say that the genome of the cell is over here, and let's just make it a bit smaller. Let's say this is the genome. So this genome is going to be replicated, and this genome is going to be given to this bud. Okay, so now this bud, I'll just make it a bit smaller. this bud is going to have this entire copy of the genome. Then this genome is going to enlarge. So if it's this big, it's going to slowly grow, and then it's going to be separating from the parent cell. That is what is happening. So do you guys see how over here it's just in the randomly just attaching to the cell membrane and then dividing, but over here there's this bud that forms peripheral to the main functions of the cell, and then it slowly just buds off, budding. Okay, so just one word that I want to write here that I think is important for you to know. The rest is pretty simple. That is protuberance. Okay, so it's just a word, and just know, okay, this word, if they're using the word protuberance, it just means it's a thing, it's a vesicle, whatever, I don't care. It's a thing that protrudes from something. In my O-level classes, I say that if you know English, you can easily get an A in biology. And that's true over here as well. So protuberance is just an English word. Just keep this thing in mind, and hopefully it will help you guys a lot. Now let's move on to our sexual reproduction. Okay guys, let's move on to the sexual reproduction, and I write everything down in steps, but this thing, the binary fission, the budding, it's really simple, so I haven't written any steps over here. So if you guys think that is not helpful, if you guys want me to list everything down like I've been doing in the previous videos, let me know and I will do that. Okay, so the most important thing, the most high yield thing to know is that there is going to be no meiosis or fertilization. Okay, get that idea out of your head, that it's going to be like normal sexual. Over here it means a complete different thing. It basically means that there's going to be a transfer of genetic material. GM means genetic material from a donor cell, how do you spell donor? No, that's the donor that you eat. This is the donor, right? Okay, from the donor cell to the recipient. I don't know the spellings of this either. I think this is right. I don't know. Okay, recipient cell. Okay, this is given a name, this thing. They might ask you, okay, what is the definition of defined sexual reproduction bacteria, or they might ask you, what is genetic recombination? Guys, if you have done your gene tech, you guys probably know what genetic recombination is. This is pretty much that thing, but this obviously is happening naturally. And there's another word for it that I want you guys to keep in mind, and that is parasexuality. Okay, parasexuality. All right, there's three ways. It's not a high yield thing. It's not a very high yield thing for you to know exactly all about the different ways. There are a couple of channels that went into a lot of detail, and when I was just looking for material, but I don't think this is important because it is not mentioned in a lot of detail. So I'll just stick to the amount of detail that is required for you to give your exams. Okay, so three different ways. First thing, keep that thing in mind: conjugation, transduction, and lastly we have transformation. Okay, it's pretty simple, and I'll try and explain this to you guys and not make you guys memorize this because I hate that. Okay, so conjugation is only going to be occurring in related bacteria. Okay, it can't happen in just like two random bacteria. It usually happens in related bacteria. In transduction, we have a third party that is involved. For example, you guys probably all know about bacteriophages. These are small bacteria that these are small viruses that affect bacteria. So over here this is involved, and transformation generally is done by unrelated bacteria. Like related bacteria can also do it, but generally unrelated bacteria. So when there's sexual reproduction between unrelated bacteria, it's mostly due to transformation. Okay, so now let's talk about conjugation. So what happens is, let's say there's one cell, and then let's say there's two cells. This is the second cell, sorry. Okay, so now I have this structure formed in the middle, which is called a conjugating bridge. Okay, a conjugating bridge, or the word that they want you to know is sex pilus. Okay, so this is a thing that is formed, and now what happens is, what plasmids are, right? These are these small tiny circular pieces of your genetic information. Now this basically travels, it can be exchanged, it can be donated, or whatever. Okay, so it goes from one cell to the other cell. Okay, so that's pretty much it about conjugation. One thing that I do want you guys to keep in mind is that even though it is related bacteria, it is different mating types. I wouldn't use the word genders for it or sexes for it. A better word is just different mating types. And to explain to you guys, just imagine this is a male, this is a female. This is a very big oversimplification, but this is pretty much the detail that you are required to know. Okay, so conjugation, different mating types, related bacteria. Just one thing, plasmids. Okay, plasmids are the primary thing that we use. It's the primary organelle that we use. Organelle is a bad word. It's a primary actually it's just the primary thing that we use in conjugation. Okay, so moving on to transduction. So in transduction, what happens is bacteriophage, essentially you could think of it this way, that what the bacteriophage does is that it's going to be stealing portions of the bacterial genome. So it's going to be sort of stealing these portions. Okay, and once it goes someplace else, it is going to be essentially, upon a new infection, it is going to be giving it's going to be giving these genes to other cells, to new cells. Okay, let's just try and understand this. There's no point in memorizing it. Let's just try and understand this. Okay, so two cells. Okay, what's happening is they can be related, they can be unrelated, we don't really care. So what's happening is that this is the genome of this bacteria, right? And this is the genome of this other bacteria. So now what the virus is doing is that it's here. Okay, it sort of stole this portion. Okay, let's say just sort of stole this portion. So let me just draw it. It stole this portion, and then what it did was that it overtook the entire cell. It broke down the entire cell, and how a virus replicates and all. And then let's say now the cell membrane is disintegrated. Now what the virus does is that the virus, so let me just quickly draw the virus. So the virus is carrying this thing, it's carrying this DNA. Okay, again, gross, it's a huge oversimplification, but I just want you guys to really try and understand this. Okay, understand what's happening. So the bacteria, so the virus is now here, and the virus, what it does here is that it's going to sort of insert this genome in this new cell. Okay, and this is how the entire process of transduction takes place. Okay, let's move on to transformation. So in transformation, it's completely unrelated bacteria, and it's pretty simple. Let's just say that this is a piece of bacteria, and this is piece of bacteria, it's a bacterial cell, and let's say that this has all of these different plasmids. Okay, now what happens is that there's death, that this cell dies. Okay, or in certain forms of bacteria, what they do is that they intentionally push these plasmids out. So they sort of make these gaps in their cell membranes, and they push these entire plasmids out. So these plasmids, let's just say, okay, now the plasmids are out. The plasmids, they move out, and now they might be taken up through diffusion or through other different ways into different cells. Let's say 3 went here, 3 went floating, went all the way here, and this one went all the way inside this cell. Okay, so this is what happens. This is the 3 different types. I used a bit of a different approach in this video. Instead of listing everything down, asking you guys to memorize it, I used the approach of teaching that I use in my classes. My classes aren't really that well planned. I just try and teach that concept. Planned not in the sense, these videos, they're scripted, so my classes aren't scripted. So like today's video was not scripted. I hope you guys liked it. It was different, and if you guys want me to go back to the previous way जहाँ पे मैं एक चीज़ को ऐसे करके लिखता था, and I would just ask you guys to memorize it, I can also go to that, but I just think that this might be a bit better. That's all for today, and thank you so much, guys. And I'm sorry if I made like a couple of mistakes like bolte हुए, because I'm just really tired, and my neurosciences module is going on. But that's all, and thank you so much, and hope to see you in the next video. Allah Hafiz.